Hello, and welcome to this session on LO3, Understanding the Mode of Action of Enzymes. Today, we will explore how enzymes regulate the rate of chemical reactions in living organisms. Enzymes are highly specialized biological catalysts that are fundamental to metabolism, enabling reactions to occur at rates necessary for life. Without enzymes, many biochemical processes would be too slow to sustain cellular function. In this session, we will examine the structure and function of enzymes, how they facilitate reactions, and the various factors that influence their activity. By the end, you will have a clear understanding of the mechanisms by which enzymes control and optimize chemical processes within living systems. Enzymes are proteins composed of amino acid chains linked by peptide bonds. Their function relies on their structure, including primary, secondary, and tertiary levels. The tertiary structure forms an active site, crucial for enzyme function and specificity. Any alteration in this structure can hinder enzyme activity. Understanding these structural levels is key to grasping enzyme action. Enzymes act by lowering activation energy, enabling reactions to occur at body temperature and pH. They form enzyme-substrate complexes to stabilize transition states. This catalysis is vital for metabolic reactions. Enzymes facilitate both catabolic and anabolic reactions. Each step in these pathways requires specific enzymes. This is the main call-out for the scene, highlighting the core idea. Enzymes play roles in catabolic and anabolic reactions. Catabolic reactions break down molecules, releasing energy, like glucose breakdown in respiration. Anabolic reactions build molecules, requiring energy, such as protein synthesis. Different enzymes are necessary for each metabolic step. This specificity is crucial for efficient metabolism. The lock and key model describes enzyme specificity. The active site has a fixed shape matching the substrate. Only the correct substrate can bind, forming an enzyme-substrate complex. This explains why enzymes are highly specific. However, it assumes the enzyme structure remains unchanged during the reaction. The induced fit model suggests a flexible active site. Upon substrate binding, the enzyme changes shape to fit better. This enhances enzyme-substrate interaction and lowers activation energy. The model accounts for enzyme flexibility and explains bond strain. It's a more accurate representation of enzyme action. Temperature influences enzyme activity significantly. Higher temperatures increase molecular collisions and reaction rates. Beyond the optimum, enzymes denature, altering active sites. This reduces substrate binding and slows reactions. Understanding temperature effects is crucial for enzyme function. Each enzyme has an optimum pH for peak activity. Deviations disrupt bonds, maintaining enzyme structure. Extreme pH levels can denature enzymes, altering active sites. This affects substrate binding and reduces activity. Examples include pepsin in acidic conditions and intracellular enzymes at neutral pH. Substrate concentration impacts reaction rates. At low concentrations, increasing substrate boosts reaction rates. Eventually, all active sites become occupied, saturating the enzyme. Further increases in substrate concentration don't affect the rate. Understanding this helps optimize enzyme reactions. Inhibitors reduce enzyme activity by interfering with substrate binding. Competitive inhibitors compete for the active site, while non-competitive inhibitors alter enzyme shape. Substrate concentration can mitigate competitive inhibition. Non-competitive inhibitors affect activity regardless of substrate levels. Understanding inhibitors is key for controlling enzyme reactions. The lock and key and induced fit models explain enzyme action. Both describe enzyme specificity and substrate complex formation. The lock and key model assumes a rigid active site. The induced fit model accounts for enzyme flexibility. Understanding both models enhances comprehension of enzyme mechanisms. The lock and key model features a fixed active site shape. Substrates fit precisely, forming enzyme-substrate complexes. This model explains enzyme specificity but doesn't account for structural changes. It assumes enzymes remain unchanged during reactions. This limits its explanation of activation energy reduction. The induced fit model presents a flexible active site. Substrate binding causes conformational changes, enhancing fit. This model explains enzyme specificity and activation energy reduction. It accounts for bond strain and substrate manipulation. The induced fit model is widely accepted for enzyme action. Both models agree on key enzyme features. Enzymes bind specific substrates at active sites. Enzyme-substrate complexes form during reactions. Enzymes are reusable and not consumed. Specificity arises from enzyme-substrate interactions. These similarities are foundational in enzyme studies. The models differ in active site flexibility. Lock and key assumes rigidity, while induced fit allows shape change. Induced fit explains activation energy reduction better. It accounts for enzyme flexibility and bond strain. These differences highlight the induced fit model's scientific acceptance. Environmental conditions affect enzyme reaction rates. Factors like temperature, pH, and substrate concentration are crucial. Changes alter enzyme-substrate complex formation. Enzyme concentration and inhibitors also play roles. Understanding these factors is vital for enzyme-controlled reactions. Temperature affects enzyme and substrate kinetic energy. Increased temperature boosts collision frequency and reaction rates. Beyond optimum temperature, enzymes denature, altering active sites. This reduces substrate binding and slows reactions. Understanding temperature effects is crucial for enzyme function. Each enzyme has an optimum pH for peak activity. Deviations disrupt bonds, maintaining enzyme structure. Extreme pH levels can denature enzymes, altering active sites. This affects substrate binding and reduces activity. Examples include pepsin in acidic conditions and intracellular enzymes at neutral pH. Substrate concentration impacts reaction rates. At low concentrations, increasing substrate boosts reaction rates. Eventually, all active sites become occupied, saturating the enzyme. Further increases in substrate concentration don't affect the rate. Understanding this helps optimize enzyme reactions. Increasing enzyme concentration raises reaction rates. More active sites are available for substrate binding. Reaction rates rise proportionally if substrate is not limiting. Enzyme concentration is crucial for optimizing reactions. Understanding its impact helps control enzyme activity. Inhibitors reduce enzyme activity by interfering with substrate binding. Competitive inhibitors compete for the active site, while non-competitive inhibitors alter enzyme shape. Substrate concentration can mitigate competitive inhibition. Non-competitive inhibitors affect activity regardless of substrate levels. Understanding inhibitors is key for controlling enzyme reactions. In conclusion, the induced fit model offers a more accurate and comprehensive understanding of enzyme action. It emphasizes the flexibility of enzymes and their ability to lower activation energy, providing a realistic depiction of how reactions occur in living cells. While the lock and key model serves as a helpful introductory concept, the induced fit model better reflects the dynamic nature of enzyme function. A thorough understanding of enzyme mechanisms not only deepens our knowledge of fundamental biological processes, but also underpins important scientific and medical advancements. Any questions? Contact us on WhatsApp at 07484361688. We're here to support you. Thank you for your time and see you at the next session.